Okay, so it's my pleasure to chair this first session of the main meeting. And so this session is facilitating access to data. And of course, this is the underlying core principle, I think, of Global Alliance is facilitating this access. There are obviously many challenges associated with access to data. And I think all the questions that have come up have highlighted some of those challenges. But at the Global Alliance is, I think, gone a long way towards overcoming some of these challenges through their clinical, regulatory and ethics, data, and all the other working groups, and particularly data. So Phil put up the FAIR principles, and I think the data working group, along with some of the ethics groups, have gone a long way towards addressing the accessibility, the interoperability, and the reusability of data. So this session is three talks, and they are basically dealing with different aspects of facilitating access to data. So I'd like to introduce this first speaker, who is, Serena Scollen from the ELIXIR project. So she is the head of Genomics and Human Genomics and Data Translation, or Translational Data. Thank you.
So I'd like to start my talk off with a little bit of a vision about where we'd like the human genomics field to be going, really. And I think it would be really game-changing if we can achieve a much bigger vision for the human genomics field. So the data challenges have changed over the last decade. It's no longer a challenge to generate data. It's now quite cheap and quite quick to generate even whole genome sequence data on a number of individuals. So what is then important is how we link that data. So how can we put together an infrastructure where we can manage, archive, share, and reuse that data effectively to get the scientific and medical community on a path that is really going to be effective for discovery? So if we think about the data that we have just for an individual, we have now whole genome sequence data or other sequence data, and that can be linked to all sorts of other types of data. So omics data, proteomics, we've got electronic health record data, and now lifestyle data from wearables that are far more common for people to be collecting data themselves, as well as self-reported data. And then further from that, we have the option to have biosamples taken that can then be used for all sorts of experiments. So for example, induced pluripotent stem cells. And we're actually not very good at getting all of that to get data together for just one individual, because that data is generated from all sorts of different types and all different types of sectors, and we're not very good at putting that all together for an individual. But what I think would be really important is to get that data together for an individual, but also together and linked across over 1 million individuals. And then I think we'll be in a position that we can really get the scientific and medical community on a path to use that data effectively and expedite discovery. So what's important? We have the data. How do we find it? So we need to have platforms available that we can actually use to discover data effectively. And we need to be able to do that quickly. It's no good if it takes us months and months to get that data, because whatever we're trying to do is either gone past, your PhD student has actually moved on, or for drug development, for example, the program's actually moved on, and having additional human genomics data is actually not very useful at that point. So let me give you an example of a gene in my past research. So SCN9A is a favorite gene of mine. It's associated with extreme pain disorders. So it's Mendelian disorders. So loss of function mutations were found in 2006 that caused a congenital insensitivity to pain, so a complete loss of pain. But actually, prior to that, 2 years before, there were associations found in primary erythromelalgia. So these are extreme gain of pain phenotypes where people have extreme pain in their hands and feet. So it actually took probably another 5, 10, 15 years on top of that initial first discovery to really get to the bottom of having a compelling package of data. So with the additional functional data and with having additional families come forward and mutations identified. And now there is a package of data and it's a very appealing drug target based on this human genetic evidence. But it took a long time. So anything we can do to put together that data that can then expedite that discovery and get us to the point where we have a compelling package of data is really, really, really going to be useful and really going to help us move forward and transform the health and scientific discoveries that we can do to date. So I'm from ELIXIR. We are an intergovernment organisation that's been put in place to put together infrastructure for the life scientists across Europe, but of course we also have to work internationally to really use data effectively. So we are organised around 5 platforms: data, interoperability, tools, compute, and training. And we have currently 4 use cases, 2 of which fall into the discussion today: human data and rare diseases. We're organised as a hub and node structure. So I'm based in the coordinating hub in Cambridge in the UK on the Hingston site, and we have nodes in each of our member countries. So these are national bioinformatics centres, where each of those nodes has their own specific national priorities and national strengths. And we work to bring all of that together with all of the experts and all of the data, and have access to over 160 institutions through the system to be able to then coordinate an infrastructure that becomes able to scale with the challenge. So for example, the challenge I initially set out of trying to pull together more than 1 million people. So these are the current members. We have 20 members, so 19 countries, and EMBL is a European node, and we have 1 observer, Greece. So what are we doing with the Global Alliance? So we are coming together with the Global Alliance and the user community to develop standards and tools. So we have to do this as a community, and it's only then that we'll be able to simplify the way that we can search for and request identifiable data across international and also national genomic data resources. So I'm really pleased to be talking about a project that started well before I joined ELIXIR, the Beacon project that I'll be now co-leading with Mark. It's a public data discovery web service. So it's a very simple concept, and it was really established as an experiment to see if people would share some level of data, and it's been successful. So all it is a simple web service where you put in a request to a data cohort or an institution to say, do you have information about the allele at a specific chromosome at a specific position? And all you get back is a yes/no answer. Has that allele been found? And then further than that, you have the possibility of asking across a whole network of beacons. So you can ask all of the institutions that have their data available, is that allele present in your data set? Yes or no? So this saves an enormous amount of time by actually identifying cohorts that have your data, rather than having to contact the PI of each institute and find out if they have a specific data set. And you can then go on, obviously, and find out if they have the phenotype you're interested in or what the contract is like for actually accessing that data. This is what it looks like, a simple web service. And this is what you get back. So a number of institutions that have the allele that you're interested in. So this is actually an example that I put in from, again, previous research in pain genetics, which is an allele in a potassium channel gene, KCNK18, which encodes for the potassium channel TRESC. Now, this is an interesting gene because there has been a private mutation, funnily enough, in a Canadian family that has been identified to be associated with migraine with aura. And that was published back in 2006. But following that, there's been no other reports of that loss of function mutation being found to cause migraine. So this is a nice example of where you really need to be able to access the other variants to really try and establish if there are associations with neurological disorders, to put that package of data together to make a compelling case, for example, for drug discovery, to say, here's a nice gene that we can then follow up and develop therapeutics to treat neurological disorders. So this is actually another loss of function variant in the KCNK18 gene, and there are several of these. So you could put however many loss of function variants there are, 5 or 6, into this search engine and say, actually, where is your data that you can then follow up and find out if they have phenotype data that you could then use to study. So what's ELIXIR specifically doing with the beacons? Well, we're funding an implementation of the beacon technology within the ELIXIR nodes. So what we're able to do is bring that center of European data to the beacons. We also have developed a 3-tiered access system for accessing data at different levels, so whether it's just public data that's accessible to anybody, or whether you have data that's accessible to a bona fide researcher that you've identified through certain parameters. And then, of course, the controlled data that needs a signed agreement as well. And if you want to find out more about this, there is a website link there. So the next steps moving into the next year will be to monitor the security, deployment, and look at data sources. So it's important for security. It's possible to re-identify people. If you have the same sequence, you could actually amass data by putting in a number of different requests. So obviously we need to make sure that that's monitored and that we prevent people re-identifying who particular people are through this simple mechanism. We're also looking at deploying the beacons. We're packaging the beacons up so that the ELIXIR nodes can each light a beacon if they have data that's accessible. And we are also looking at nodes or European data that's outside of that and partnering with national data owners as well. Further than that, we can then consider looking at making other data available. So, for example, if we can link up with phenotype data and make that as part of the beacon, and that will be further stages that need to be discussed further and been discussed over the last couple of days. So we're hoping to maintain this key partnership with ELIXIR and Global Alliance. It's very important to make this international, not just European, from our perspective. And it's then that we can look at really increasing the genomic data sharing and facilitate sharing. So it's a small answer in some ways. We have to look at this as a chunk by chunk. There's going to be many problems we need to solve. It's not simple to get an infrastructure in place, but I find it really exciting because once we have this infrastructure in place, we're really going to be able to start to understand disease more fully. We're going to be able to develop pharmacological hypotheses, and we're then going to be able to look at patient stratification and precision medicine approaches by putting some of these solutions and standards and tools in place. So to give you an example, you might think it's a stretch to say it's precision medicine approaches, but another example, going back to my original NaV1.7 SCN9A gene example, here there are NaV1.7 blockers in development by several companies, and each of those companies have tried to access the primary erythromelalgia cases that have a gain of function in this gene to use those individuals as a proof of mechanism that the drug is working. But actually, they're very, very rare patients, so it's very difficult to find them. So there's another use for the beacons that I can see is that you could put in the variants that you're interested in testing and essentially get a list back of where they are across the world. And that's a great way of actually really reducing the time it takes to set up a clinical trial and test a drug for proof of mechanism. So there's many, many uses that I think the beacons will be really, really valuable for. And I think really then once we put that all together, we'll be able to unleash the possibilities for genomics and health in the future. Thank you.
Floor is open to questions. You want to go to the microphone? Let's just go to the microphone. Yeah?
Is there any possibility to look for other variation at that same position? So in other words, it may not be just that specific nucleotide change, but something else that alters that nucleotide in beacon.
So you can put in what?
So instead of a C to T, if it's a C to G or...
Yeah, absolutely. Yeah,
okay.
You can put in whatever request you want, yeah.
Okay, and not gene specific? It's
just
too wide.
On any specific position, you can put in a search.
Oh, so it's, you can be quite broad in your search terms.
Yeah.
So I have a question. You alluded to the parameters used for registered access for what is a bona fide researcher. Can you elaborate on how that's implemented?
Yeah, so there's a lot of discussion around that in the last couple of days. So there's various ways of doing it, trying to identify, and it would be different levels for different data sets. So for example, you could use your ORCID ID and use that as a mechanism of identifying where you are. But there's a number of parameters that still are in a lot of discussion about how we do that effectively and how we can actually, what is a bona fide researcher. So there's more work to be done there.